I'd like to now talk about sea urchins and tunicates with respect to their embryonic development. Sea urchins and tunicates are deuterostome invertebrates. You may recall that deuterostomes are animals in which the blastopore becomes the anus during development, as compared to protostomes where the blastopore becomes the mouth. If you look at this diagrammatic representation of some of the groups of animals in these categories, you can see in particular in the deuterostomes, you can see there are echinoderms and sea urchins. Starfish would be a part of that. There are hemichordates, the arrowworms, cephalochordates, tunicates, and urochordates. We'll talk about two examples. We'll talk about sea urchins, which are echinoderms, and we'll also talk about tunicates, which are urochordates. Keep in mind that the deuterostomes also include all of the vertebrates, and later on we'll be talking about all of the vertebrates in some detail with respect to their embryonic development. So to begin with, let's talk about the sea urchins. Sea urchins have been exceptionally important organisms in studying how genes regulate the formation of the body. In fact, sea urchin embryos provided the first evidence that chromosomes were needed for development, that DNA and RNA were present in each animal cell, that messenger RNAs directed protein synthesis, that stored messenger RNAs in the ovum provided the proteins and RNAs for early embryonic development, that cyclins controlled cell division, and in fact, the first cloned eukaryotic gene encoded a sea urchin histone protein back in 1975. So, sea urchins have been an important animal in embryonic development for a long time. With the development of new genetic techniques, sea urchin embryos continue to be major organisms for elucidating mechanisms by which different cell fates are specified. Let's look now at sea urchin cleavage. Cleavage in the sea urchin is holoblastic. This is a type that happens in eggs with sparse yolk, and you'll note that in the sea urchin there's very, very little yolk, and that yolk that's present is spread throughout the egg. So the cleavage furrows initially extend through the entire egg, north-south, so we call this meridional cleavage, and the first two cleavages are perpendicular to each other, so you end up with 4 cells that are meridionally organized as far as cleavage goes. Then the next cleavage division, we get an animal half and a vegetal half, so that's an equatorial cleavage. From there, some very interesting things happen. The top half will have meridional cleavage, whereas the bottom half has equatorial cleavage, but it's not equal. It forms macromeres and micromeres. Certainly cleavage continues until you get a large number of cells, but if we look at this example, this is really at the 8-cell stage. It's this stage now dividing into this stage, forming into the 16-cell stage. We see that here on the top we are getting meridional cleavage. The cells are dividing to form this way, and then in the vegetal half we're getting equatorial cleavage to form the larger macromeres and the smaller micromeres, which are down here on the bottom. If we now look at this with Nomarski optics or differential interference contrast optics in the light microscope, these are photographs of living cells. You can see here is the fertilized egg, and the blastocele will eventually form a coelomic cavity. Let's go through this. We get fertilization taking place. The black arrow is the site of the sperm penetration. The white arrow is where the vegetal pole is in the cell, and here on the outside we have now a fertilization envelope to keep further sperm from penetrating into the egg now that that one sperm has gone into the substance of the egg. Here we get the first cleavage division. Again, the fertilization envelope is there. Now we go to the 8-cell stage. At this point, in further stages, when we get to the 16-cell stage, which is right here, you start to get a blastocele forming. This blastocele contains proteinaceous fluid inside. It continues to develop until you get to a fairly large number of cells. I should mention that the micromeres, of course, are smaller. They do not continue to divide. Eventually, as the other cells divide, all of the cells become the same size, because the micromeres now will sort of fit in size-wise with the others because the others have continued to divide. The hyaline layer is present on the outside, and it will form a future vegetal plate. In addition, if you look at this blastocele, the animal hemisphere is over here. The vegetal hemisphere then is over here, where you can see that light line. Tight junctions connect blastomeres into a seamless epithelial sheet that completely encircles the blastocele. As the cells continue to divide, the blastula remains one cell layer thick, thinning out as it expands. So that's really only a single cell layer that continues to get thinner. Rapid cell cleavages last through the 9th or 10th division, depending on the species, and by this time the fates of the cells have actually become specified as to what they are going to be. Thus there is an apical outside of the cell and a basilar inside of the cell. Apical outside, basilar inside. So there's a polarity of the cells by that point in time where you have this blastocele formed. On the outside, cilia form, so there's a ciliated outer layer as well. The cells at the vegetal pole begin to thicken, forming a vegetal plate. The cells of the animal hemisphere synthesize and secrete a hatching enzyme, which will eventually break down the fertilization membrane, and the blastula is able to hatch and come out into a free, ciliated swimming state. By the 60-cell stage, most of the embryonic cell fates, as I mentioned, are specified, but the cells are not irreversibly committed. In other words, particular blastomeres consistently produce the same cell types in each embryo, but these cells remain pluripotent and can give rise to other cell types if experimentally placed in a different part of the embryo. If we look now at the fate map and cell lineage of the sea urchin, we can see that there's animal half, and that will give rise to the ectoderm up here. Forms the ectoderm, the larval skin. It will form the neural tissue, just like in other organisms we've looked at where the ectoderm or epidermis involutes to form the nervous tissue. The vegetal layer, there are a couple of vegetal layers. There's a veg 1 and a veg 2, as you can see. The veg 1s are the cells that enter larval ectodermal or endodermal organs, and veg 2 cells that populate the endoderm, coelom, and non-skeletal mesenchyme, such as pigment cells, immunocytes, and muscle cells. The micromeres, there are two categories of micromeres. There are the large micromeres. They're the first tier. They're the darker red color there. And you can see that they're distinct but much smaller in terms of diameters than the cells that are up in the animal pole or the other cells in the vegetal pole. What these micromeres do, the large micromeres, is to form skeletogenic mesenchyme that forms the larval skeleton. The micromeres really play no role in embryonic development per se, but later on they will help to form the germ cells. So they will form the eggs and the sperm later on as the animal continues to differentiate and form into adulthood. There are some interesting experiments that tell you what these micromeres are capable of doing. If we look at the panel A group, we can see this is normal development in which you have the animal hemisphere and vegetal hemisphere and micromeres intact. They will form into a blastula and then a pluteus larva and then into the adult organism. If you take the animal hemisphere alone, however, it will simply form into what's called a dower blastula. It just forms a layer of cells which are ciliated, and these layers of cells then will be totally animalized. However, they will not really form into a functional kind of animal of any kind. If, however, you take those animal hemispheres and you add micromeres to it, these micromeres will basically induce this animal hemisphere to form into a definitive, recognizable larva. It looks very similar to the pluteus larva, and what it's been able to do is to take cells from the animal hemisphere and even make endoderm-type structures from them, keeping in mind that the endoderm was totally and exclusively a component of the vegetal pole. There is no vegetal pole here. However, these micromeres are able to convert animal pole cells into vegetal pole derivatives, in particular endoderm, that will be from the animal layers. Another interesting thing about the micromeres is that they really are the organizers. They're the animal organizer. They're very similar in function to the dorsal lip of the plastipore that we talked about in amphibians, which would initiate the formation of an animal during gastrulation. These micromeres do essentially the same thing in sea urchin embryos, and these experiments would certainly demonstrate that. The micromeres will induce a secondary axis. As you can see here, if you put the transplanted micromeres on the animal pole up here, they will induce the formation of a second animal, basically. Skeletogenic mesenchyme forms, just like it did from the endodermal area down below. These transplanted micromeres then induce the endoderm gene expression in these ectodermal cells to cause them to form into animal pole cells. And this development will continue, and you will get skeletal rods, and you will get all of the structures that will lead to the formation of a pluteus coming from each end, top and bottom, of these developing micromeres. If we look at sea urchin development following the fate of the cellular layers of the blastula, if you look at A, there's a fertilized egg, animal pole, vegetal pole, and then it undergoes divisions into the ectoderm, endodermal areas, the non-skeletogenic mesoderm as well as the skeletogenic mesoderm. It will end up forming a blastula with a ciliary tuft. As this progresses, it begins to invaginate, and the endoderm and gut forms down here in E. There is skeletogenic mesenchyme cells and non-skeletogenic mesenchyme cells forming the invaginating endoderm, which will form the gut, as I mentioned, and it is now really a bilaterally symmetrical embryo. With continuing development, the anus area grows up, forming the gut. So here's where it gets its name, deuterostome. The mouth forms second. So the anus forms first. The mouth hasn't quite formed yet, but that will form then second and lead to the formation of the embryo. This is called a prism stage larvae here. Here you can see a little more advanced version, which is approaching the pluteus larvae stage. This illustrates differential interference contrast microscopy of living cells that are undergoing differentiation from one of the sea urchins. And if you follow at the different time frames, you can see that this is developing fairly quickly, from 9 hours to 18 hours, going from the upper left corner to the lower right corner. You can see that blastulation is taking place. There's a blastocele formed. You're starting to get the blastopore, which is forming the gut, and this gut will extend up into the digestive tube that will lead to the formation of the pluteus embryo after about 24 hours. This shows the invagination of the vegetal plate. Here you can see that the blastocele again is forming, the blastopore is forming. This is a scanning electron micrograph. The blastopore is visible in the vegetal plate of this quite early gastrulous stage. The fate map viewed from the side and looking upward, this shows the vegetal surface, and on the right you're looking into the blastopore. So it gives you a fate map of where these things will end up. The mesodermal structures going inside are in the center part of that blastopore, and the pink areas, and then the yellow areas, the foregut, midgut, and hindgut, will appear as concentric layers, but as development progresses, these will extend out and basically form those areas of the digestive tube. Of course, the blue ectoderm stays on the outside and will eventually cover the entire animal. This shows cell rearrangement during extension of the archenteron in the sea urchin embryos during early gastrulation. You can see that the blastopore is coming up, and the interesting thing is that wide in diameter at that point, because there are a number of cells that go around the diameter. However, as it elongates and moves toward the animal pole, that is this developing gut area, this gut thins out, becomes more of a tubular, smaller tubular structure that will extend and form a smaller diameter tube. And if we look at that again with differential interference contrast optics, you can see that kind of a relationship, and also as development progresses, you are seeing this pulling of the gut tube up toward the animal area of the developing embryo, and you can see strands that are hooking up the inner surface of those. And as you look at the extension of these fibers will extend up and hook on to the inner surface, and it will pull up this gut tube to get it in position. And the archenteron, then, the inner part of the gut, is forming. So the filopodial extensions of non-skeletogenic mesenchyme cells extend these filopodia, then, from the tip of the archenteron. The Pluteus larva, then, in order to reach the next stage and to become an adult, has a crucial step that it must do, and that is the asymmetric formation of the imaginal rudiment on the left side of the bilateral Pluteus larvae. So as you go through the top panel here, A, you can see that the egg and the blastula show radial symmetry. Then, once gastrulation starts, and through the Pluteus stage, you get a bilateral symmetry. And this is what makes these organisms related to some of the higher vertebrates, in the sense that during this embryological stage, it shows bilateral symmetry, similar to what the vertebrates, many of the other deuterostomes, show. Here we see during gastrula and the Pluteus stage, it's bilaterally symmetrical. And then as you get to the Pluteus stage, you can see that there's an imaginal disc, an imaginal rudiment that forms on the left side of the embryo. And as it does so, it continues to develop. So if you look in B, you can see that it's an imaginal rudiment that has a five-fold radial symmetry. So it's a pentaradial symmetry. The bilateral symmetry during development, though, as I mentioned, shows the evolutionary connection to bilaterally symmetrical species. For this organism to develop into an adult that we see in the upper right corner, this disc, this imaginal rudiment, this disc, will basically become free from the rest of the embryo. In fact, it will shed the rest of the developing embryo, and this will fall to somewhere in the bottom of the sea. It will attach to something, and it will become sessile for the rest of its existence. It will no longer be motile and be able to swim around. It will now basically anchor itself to some structure, and it will remain as an adult organism, functioning as a sessile organism somewhere in the ocean. This completes really what I wanted to talk about with respect to sea urchins. We will now move on to another deuterostome invertebrate, the tunicate, or sea squirt. So let's now talk about sea squirts with respect to their development. You can see that if we look at the fate map, panel A, this is an invertebrate chordate. They lack vertebra, but they have a notochord and dorsal nerve cord when they are at the free swimming stage, very similar to a tadpole in some of the vertebrate embryos. In going through the figure, you can see also that this particular, if you look at the fate map, it's not too unlike what you would see, let's say, in an amphibian fate map, where you've got ectoderm on the top, there's neural ectoderm that will form, there's a notochord right below the neural ectoderm. All of these are the same relationship as you would see in an amphibian embryo, or a bird embryo, or a mammal, or a reptile, or whatever, in terms of vertebrates. Here we see the endoderm at the bottom, muscle tissue, and then as it continues to divide, here we're at the 8-cell stage, we can see that it forms a vegetal pole as well as an animal pole on the top, and it becomes bilaterally symmetrical with a separation of the different components. And this bilateral symmetry continues for a number of stages of development, in fact, until it reaches adulthood, and then it loses that bilateral symmetry again. But let's look then at the cytoplasmic rearrangement of the fertilized egg in the tunicate. Here we can see, if we look at an unfertilized egg, that we have the oocyte nucleus at the top, there's a gray cytoplasm, there's yellow cortical cytoplasm that will become interesting because it ends up forming muscle later on in the embryo. And once fertilization takes place, the sperm nucleus will set up, and the entry of the sperm will set up a cascade of events that take place, including the migration of the cytoplasmic material toward the sperm pronucleus. The nuclear material will stay at the top, but then the sperm pronucleus will migrate up in that direction as well. The yellow cytoplasm will stay at the bottom and condense, and then it will separate into yolk material, a yellow crescent, and the clear cytoplasm. Events take place once fertilization occurs. If we look at this sequence and go beyond this sequence of events, we can see this happening in an actual photograph. Here there's a cytoplasmic segregation of the egg of one of the tunicates. The yellow crescent is in the vegetal pole. There you can see in B. It becomes segregated into the B4.1 blastomeres that give rise to muscle cells. So you can see that it's continuing, and as development progresses, that yellow color then can be followed throughout development, and the cytoplasm will turn into cells that will give rise to muscle tissue. Now, once you get over to the E and F stages, you start to recognize that these tunicates have certain similarities to, let's say, a frog embryo developing, where you can see a head region, you see segmented muscle that's made out of yellow cytoplasm, the yellow crescent, and that's how initially these fate maps were made, with simply the natural colors in these tunicates. As development progresses, an embryo that's eventually can be free swimming will form. We can also look at the autonomous expression of the morphogenetic factor MACHO1. MACHO1, you may recall from one of the earlier lectures, promotes the formation of muscle tissue. It's a morphogenetic factor, and in this case it's localized by in situ hybridization in the vegetal pole of the cytoplasm in the same place where the yellow crescent has formed. So this morphogenetic factor is associated with the yellow crescent that forms muscle, and so this is consistent with this being a muscle-forming morphogenetic factor only named MACHO1. It starts in the vegetal pole, migrates up the posterior surface of the egg, and it becomes localized then in the B4.1 blastomeres, which will form, as I mentioned, muscle tissue. Let's now look at gastrulation in the tunicate, and as we look at it, again, you see similarities to some of the other organisms that we will study in the future then, and to a certain extent that we've studied in the past. It reminds us of the amphibian gastrulation. Here we see diagrammatic cross-sections in A, B, and C, and in D, E, and F we see comparable scanning electron micrographs viewed from the vegetal pole, in other words from the inferior aspect, so that we are looking at the invagination of the endoderm forming the blastopore. If we follow the deuterostome gastrulation in which the blastopore becomes the anus, then during gastrulation, first of all, there's an invagination of the endoderm, as we see in A and D. We then see an involution of the mesoderm, B and E, and in C and F we will see epiboly as well taking place of the ectoderm, now moving to surround the internal organs. The ectodermal cells, after gastrulation, invaginate into dorsal aspects of the embryo and form also a brain and spinal cord. And at metamorphosis, however, all of these things that were headed toward being a functional swimming organism are lost, because all of these break down at metamorphosis. The neural tube degenerates, the animal no longer has capability of moving, and the animal basically becomes sessile and is like the sea urchin. It becomes attached somewhere to the bottom of the ocean. However, in that time period just before metamorphosis takes place, you can see that as development progresses, this tadpole-like body forms. This is a green fluorescent protein that illustrates the notochord forming. The notochord forming and acts almost like vertebra, although this does not have really a backbone or any vertebra, but it does have a notochord and it does have a neural tube. It also has a brain on the front end of that neural tube. So it's quite obvious here that you have bilateral symmetry, and as a modal chordate embryo, complete with a brain, a neural tube, and notochord, as I said, which degenerate at metamorphosis to lead to a non-modal sessile existence as an adult. However, these obvious vertebrate-like characteristics as larvae clearly show the evolutionary relationship of these animals to vertebrates and warrant certainly their classification then as urochordates. And once this metamorphosis occurs, the animal will take on this sessile existence attached to something in the ocean. And with that, we've had a quick overview of both the sea urchins and the tunicates, the deuterostome invertebrates, and that should be interesting in terms of comparing this now to what we're going to learn about when we talk about vertebrates.